Hello, everyone, and thank you again for joining this Criterion Edge webinar. For those of you that are not already aware, Criterion Edge is a distinguished global provider in the IVD industry dedicated to helping clients advance compliance and enhance market success through expert regulatory, scientific, and clinical writing services. Today, we are presenting a webinar entitled Considerations for Companion Diagnostics, Lessons Learned, and Key Takeaways from DIA 2024. Our presenter today is Dr. Sarah Chavez. Dr. Sarah Chavez is the Director of IVD and Scientific Writing Services at Criterion Edge. She holds a BS in Biochemistry and a PhD in Molecular and Cellular Biology. Dr. Chavez brings a wealth of laboratory and teaching experience to her role, with over 25 years at the bench and nearly 2 decades educating at the university level. Specializing in regulatory writing, she has crafted extensive scientific documentation for leading textbook publishers and academic institutions. Her robust background in research and regulatory expertise uniquely positions her to lead IVD and medical device companies through the complexities of IVDR compliance and readiness. Now I'm going to go ahead and hand it off to Sarah to proceed with today's webinar, Considerations for Companion Diagnostics, Lessons Learned, and Key Takeaways from DIA 2024.
Thank you so much, Justin. So today we thought it would be a great opportunity, given that DIA just happened this past week, to introduce the effects of some of these updated regulations on folks who are embarking on the companion diagnostic process. So when IVDR took effect back in May of 2022, we all know that there were these substantial changes. And the same types of changes are also being experienced here in the US with the FDA submissions. So today we're going to focus from sort of a companion diagnostics standpoint on some of the aspects and some of the insider information that we gleaned from the 2024 DIA meeting, to share some of these key takeaways, including the focus on clinical evidence requirements and some strategies to help us keep our report writing and our data collection strategies on track. So today, our learning objectives are to really understand how these new updated regulatory requirements apply specifically to companion diagnostics and potential implications from both the IVD partner as well as the pharma partner's perspectives. We also want to describe that sort of strategic approach that we can all take to ensure compliance and potentially coordination between our IVDR and our FDA requirements if you are developing not only a companion diagnostic, but also potentially a laboratory developed test. And that will cover aspects such as our quality management, our documentation, and really sort of the overall approach that we're going to take as we're interacting with your notified body. And then finally, we wanted to address some of the common challenges that have been faced during this implementation, sort of the growth pains that all of us are experiencing as they pertain to companion diagnostics and LDTs, and look into some of the potential solutions that I see coming out of some of these themes that came about from DIA 2024. So during DIA, there were a variety of sessions, including companion diagnostics and laboratory developed tests. And what we noticed was during a lot of these discussions, there was this common thread that emerged. And that was really the need for considering strategies that we're all undertaking for collecting real world evidence so that we can better evaluate the performance of IVDs throughout their life cycle, before and after regulatory approval. And we also noticed there was a lot of information being presented on the role of AI in helping to identify, extract, and present that data through effective medical writing. So real world data or real world evidence, these are essential tools for regulatory decision making when you're in healthcare and when you're evaluating pharma or medical products. Real world evidence is gleaned from collecting and identifying this real world data that's been published. And that will include information collected outside of a traditional clinical trial setting. This data can come from a variety of sources. It can be electronic health records, insurance claims, patient registries, wearable technology data, but it can also be that published data. And actually, in recognizing the significance of this real world evidence, regulatory bodies, including the FDA, Health Canada, EMA, they're all increasingly integrating that data into the drug approval processes and promoting a more comprehensive understanding of the medication effects in everyday use. As we think about the integration of real world evidence, that sort of applies to this very first learning objective, understanding these new regulatory requirements. One of the very early feedback that we've been receiving from notified bodies is that lack of clinical evidence that's being presented with regards to the IVD partner in these applications. So real world evidence can be extremely challenging when you have a condition that is difficult to assess via a traditional clinical trial. Maybe it's something that's very rare, or you don't have necessarily a large amount of patients in order to conduct a prospective clinical trial as you'd like. And actually, in Canada, the use of real world evidence is systematically being embedded in that drug approval process and in the post market surveillance, really to help with enhancing the safety, efficacy, and accessibility of these treatments. But despite all of these benefits, there are challenges inherent with employing real world data. We don't have the benefit of having an established standardization, and the quality of the data can vary depending on the data source. So we need access to the data, but we also need a method by which we can compare that data effectively. So all of these challenges really require that we take more stringent methods to validate the reliability and the applicability of these real world evidence in our regulatory submissions. The real world evidence can and does help to supplement your data, and it reduces the need for conducting randomized control trials, but it hasn't yet replaced them as that gold standard for developing and demonstrating drug efficacy and safety. It does fill really critical evidence gaps, though, particularly from the IVD partner perspective, especially if you're talking about some of these post marketing studies where you can really assess that long term performance and the real world effectiveness of the drugs in a real world setting. So, as we're looking at this type of evidence in our regulatory decision-making processes, there are some unique considerations if you are looking at it from the perspective of the IVD partner compared to a traditional medical device. So, in the past, and what really a lot of the IVD partners are struggling with, is that randomized control trials are not typically used to clinically validate an IVD. And often, IVD partners are using leftover or surrogate samples in many instances to satisfy the requirements for clinical evidence cases. So, there is this unique opportunity to develop a sort of best practice or some tools and methodologies that help us with identifying and appraising relevant IVD data that's generated through routine clinical practice and published as such. But we need to find a way to evaluate that data with scientific rigor and meet those requirements, those necessary parameters, really, that the notified bodies are expecting to ensure quality data and to support the regulatory decision making process. The quality and reliability of the available real world data doesn't always align, though, with what the notified body is expecting to see. So that's really where we need a better way of assessing and compiling this data that we've collected from real world sources. So, going back to 2019, in 2019, the FDA published these examples of real world evidence that are used in medical device regulatory decisions. In this document, they actually describe 90 case studies for real world evidence use in medical device regulatory decisions dating back to 2012. But only 8 of those examples actually are relevant to the IVD space. So, fast forward to 2020, that's when the Medical Device Innovation Consortium published their In Vitro Diagnostic Real World Evidence Framework. And that the goal there was really to provide that additional contextual information as it pertains to the clinical validation of real world data from both a pre-market and a post-market regulatory decision-making perspective. So while the FDA guidance really addresses how that real-world evidence can be useful and valuable for the regulatory decision-making for a medical device, the IVD framework explains how to apply suitable study designs and analytical methods to relevant and reliable real-world data to produce this valid scientific validity. We need to establish scientific validity as a core principle of the performance evaluation. And this real world evidence can certainly inform or support the regulatory decision making as you are seeking to clear or approve your IVD. The benefit of this second document here is that it does discuss general considerations for study design and methodology from a routine clinical setting for regulatory decisions. So how would you deal with quality data issues if you have missed data or transparency or a lack of transparency in the data generation processes, or if there are any systematic biases that you come across? And it also helps to establish the criteria for evaluating your data in the context of that benefit risk ratio. The IVD real world evidence framework presents hypothetical and previously approved regulatory cases using real world evidence in the context of an IVD, such as when collecting clinical data for the purpose of validation could be overly challenging or potentially burdensome. So here I'm presenting a really great article. It was a 2022 review article by Baumfield Andre et al. And they actually did a great job of describing several different examples of IVD submissions that really benefited from this real world evidence, which kept coming up over and over again, especially in the context of companion diagnostics and LDTs. One case study that they specifically called out was the 2017 FDA approval of a solid tumor profiling panel, the MSK Integrated Mutation Profiling of Actionable Cancer Targets, or MSK Impact. That panel specifically looks at 468 genes for mutations using the Illumina platform. Now, MSK Impact, it's a next generation sequencing based tumor profiling test. And it was supported in large part by clinical data from electronic medical record databases of patients with advanced cancer as part of the routine workflow at Memorial Sloan Kettering Cancer Center. The retrospective analyses of those records were what provided the evidence to support their pan-cancer claim. And it helped them also for the purpose of validating a test cutoff and to provide some data on the mutation prevalence. So together, the FDA and MSK developed this sort of framework for evidence generation for both analytical and clinical performance. And those particular that framework that they used basically helped them identify how to use and develop genetic testing diagnostic technologies that meet with that increasing CLIA quality requirements. But they recognized that there was a need for more rigorous standardized process to ensure the safety and validity of these diagnostic technologies, but also acknowledged that this could potentially place an undue burden of proof on the manufacturers. So, working together and using and leveraging the benefit of all of this real world evidence really allowed them to to not ignore that evidence, to use that clinical data evidence of the various biomarker genes to support their panel and establish that scientific validity. So this original approval back in 2017 led to the consideration of whether or not pharmaceutical manufacturers can derive that sort of incremental benefit from associating their drug or treatment with an officially approved diagnostic product. And that coordination between the pharma partner and the IVD partner is really critical for establishing sort of how to streamline your submission processes. By the end of 2020, the total number of companion diagnostic assays that had been approved by the FDA had reached up to 44, and that number has clearly continued to rise. The assays were largely linked to hematological and oncological drugs for a large part. So without an accurate and reliable companion diagnostic assay, these drugs will lose their value. So really, companion diagnostic assays are high-risk devices, and the regulatory path is almost exclusively going to require this submission of one of these pre-market applications or PMAs. The analytical platforms, I think, though, as you look across different examples of companion diagnostic and LDTs, analytical platforms are diverse and they cover technologies varying from immunohistochemistry, in situ hybridization, PCR, next generation sequencing, and imaging. When you look at these different analytical platforms for different companion diagnostic assays, originally, IHC and in situ hybridization were the dominating technologies up until about 2011. And that's when we started to see the polymerase chain reaction methods moving into the field. So far, the PCR technology to date makes up the largest group of these companion diagnostic assays. And again, by the end of 2020, there were a total of 16 of these PCR-based assays that had obtained approval. And that was about 36% of all of the FDA-approved clinical companion diagnostic assays. Now, as I said, these companion diagnostic assays, many of them wind up being classified as a high-risk device, and so they fall under a Class 3 under FDA submissions, which does require submission of a pre-market application or a PMA. So compared to other types of IVD submissions, such as a 510(k), the PMA really does require the most comprehensive documentation level, and it really does help to underlie that the importance of having access to clinical performance data and analytical performance data for these companion diagnostic assays. They will directly impact the ability of that assay to be approved as a treatment decision-making tool. Several of the companion diagnostic assays on the FDA's list of cleared or approved devices are also going to fall under the realm of being a laboratory developed test or LDT. And many of those have been still cleared through the submission of a PMA. In fact, as at the publication date of this particular review article, there were a total of 9 companion diagnostic assays, mostly falling under that PCR or next generation sequencing based assays that were lab developed tests. And for each of these assays, it's clearly stated in the intended use that that assay can only be used at a single specified laboratory site. But we can all see firsthand the number of companion diagnostic assays is increasing in the past decade. They're classified as high risk, meaning that they have that higher level of evidence that's required. And we also have to ensure that we have substantial documentation for the predictive properties as far as clinical evidence. But we also want to ensure assay quality and the reliability of these assays. So that could impact why we're seeing fewer of these, that the total number of FDA approvals, there aren't quite as many as we initially thought. But if we effectively use real world evidence, the overall trend and the overall, sort of discussions around this challenge at DIA were really with regards to how to better leverage this real world evidence and what that can do to help us with this process down the line. So real world data, these are a source of the data that has a lot of information that can be used to support medical device regulatory decisions. The FDA and IVDR both are willing to leverage real world data in your determinations. And the use of real world data for regulatory decision making also supports the FDA's overall mission to help patients gain access to this high quality, safe, and effective medical devices in a timely manner. So, as we support these efforts, there has been some pending legislation in the US Senate to ensure more efficient use of the real world data and the real world evidence. But really, as we're trying to further this goal, which we all share, regulatory authorities are trying to continually improve upon and increase the efficiency of real-world data regulatory processes. As we look in the EU, the IVDR allows for a wide selection of evidence types when you can provide that due justification, and that includes real-world evidence. And in fact, IVDR requirements open up multiple avenues for the use of real-world data. They emphasize the traceability based on unique device identification, or UDI, that enables the design of product-specific studies. And there's also this emphasis on strengthening post-market surveillance requirements to ensure and encourage the use of retrospectively collected data in the interest of keeping the process moving forward. So, in fact, this real world data is a valid source of evidence to support your regulatory submissions after it's been subjected to the same level of rigorous scientific method and the appropriate level of analysis. In order to really access and really harness the power of real world data, though, we have to ensure external collaborations, and that will include public private initiatives, academic collaborations, industry partnerships, and leveraging all of the publications and being able to access the publications that are focused on these topic areas. In just this past May, the FDA published a final rule with regards to laboratory developed tests to regulate them as a medical device. And this rule had been long anticipated. There had been a lot of interest waiting for this rule to be published because lab developed tests, or LDTs, they are an IVD product that's intended for clinical use and designed, manufactured, and used within a single clinical laboratory that meets certain regulatory requirements. By regulating them as a medical device, it subjects the LDT manufacturer to these same medical device requirements. So now these manufacturers are being expected to conduct the same types of pre-market reviews, quality system requirements, adverse event reportings, establishing registration and device listings, labeling requirements, and also investigational use requirements. So, as far as the timeline goes for really pushing forward with our attempts to integrate these rules for LDTs, the beginning stage, the stage one, that's going to occur in May of 2025. And that's where so it marks the 1 year from the publication of the final rule. And that's where the FDA is going to start to expect compliance with medical device reporting requirements and correction and removal reporting requirements and QS requirements that require your complaint files. And there are other requirements during the sort of phase out or this phase out policy as they're trying to implement these different requirements over the next few years. Really, the takeaway that we're hearing from many individuals as they're trying not to panic with regards to these new requirements for LDTs is to figure out how can we be proactive and try to figure out better ways to streamline our process so that we can be collecting not only pre-market but post-market data, conducting these surveillances, and identifying data as it's being published in the literature. So, some of the key takeaways that we identified just coming at these conferences from the perspective of being the medical writer, these real world evidence, this is going to be a really robust source of potential data that's going to help us counteract that sort of common shared feedback that we're getting from notified bodies that we lack clinical evidence. The real world data helps us support the scientific validity, but it also helps us show things like the test performance in real world settings. It helps us support our intended purpose statement in all of the various clinical outcomes that we're identifying. The real world data also can do a great job of supporting your benefit risk profile and establishing the state of the art. But we also want to think about it in terms of clinical utility. How does the safety and the quality and the efficiency of this testing directly impact healthcare practices? So, in order to reach sort of our ultimate goal of writing a really robust scientific validity report, we have to have a more concerted effort to analyze this data, identify this data, and be able to make that comparison, to be able to conduct a really true, robust meta analysis of published data. Going back a step though, moving forward, the goal is really for researchers and developers to start anticipating this need moving forward. So to more thoughtfully design our real world studies with appropriate analytical and statistical methods and controls in place. But until we get to that stage, we're really left with these data sets that are sometimes difficult to compare across different publications. You may have the same device that's being um, assessed, but sometimes the data, the actual performance of the device is not necessarily the focus of the article itself. So you're sometimes limited in the um, the relevance of the data that we're left with in the published literature. So eventually, where we're hoping this will take us is that we will start to apply common data standards, common methodology, common nomenclature, and statistical methods to better assess IVD test results in a real world setting. And ultimately, that will lead us to more significant improvements in the quality and the usability of this real world data as we're pulling together our reports as the IVD perspective. Um, when we're looking at the medical writing, though, where do we go from here? As we're trying to identify and kind of scrape the bottom of the barrel, we are trying to identify all different aspects and any piece of the clinical data that has been published. How do you identify that data, and how do you then communicate that data to your notified body? So the other key takeaway we'd like to point out here is the role of AI. This came up over and over again, and you really saw so many companies and organizations proposing different integrations of AI into this whole process for medical regulatory writing. AI technologies are being utilized to automate a substantial portion of this data analysis and document preparation processes as regulatory writing goes. The automation eventually will lead us to reduce the amount of time that's required for some of these regulatory control processes. And that can ultimately decrease the time to market for things like new therapies or devices. Technologies utilizing AI can analyze large data sets from clinical trials and other studies and help us identify relevant information and generate these draft reports that hopefully, as we get further and further into the integration of these technologies, will reduce the amount of human intervention and allow for a more reliable data set that we're pulling together. So, for instance, some AI driven systems have been designed to extract specific outcomes and metrics from this sort of unstructured data. And it can be used to help you support the writing of a more sophisticated document like a CER or a PER, which are going to be critical components for meeting these new compliance standards that have been set forth by the regulatory bodies. The FDA, though, they went straight away and acknowledged the potential of AI in improving the efficiency and reliability of our regulatory submissions, and actually highlighted the role of AI in enhancing data integrity and decision-making processes. There are several undeniable advantages of integrating AI into your medical regulatory writing processes. First off, it's that efficiency. An AI algorithm can process data at a speed that a human cannot achieve. So, ideally, in the future, we're hoping that these AI integrations will allow us to reduce the amount of time we spend on manual data organization and extractions. And that sort of preliminary, that first cut of the drafting. And that's not to say that it's going to replace the need for a qualified medical writer. But what it does allow us to do is to spend more time focusing on the strategic aspects of the submission readiness rather than just doing rounds and rounds of QC to ensure the accuracy of the data that's being extracted. We can really take that deeper dive and focus on whether or not the data that's been identified through the AI algorithms is the most applicable data in order to help us support the scientific validity claims that we're making. But that improved accuracy, that I think is going to be the most exciting part of these AI tools. They're equipped with capabilities to perform consistency checks and reduce errors, and that's pivotal when it comes to regulatory writing. Some of these submissions can involve the extraction of data from hundreds of articles potentially. So the use of AI ensures that your submissions are free from these sort of common mistakes or discrepancies that could be a key source of delays in your approval process. So it helps us be more accurate and more efficient with our time. And that ultimately meets that requirement for enhanced compliance that's inherent in these updated regulations. As the regulatory guidelines constantly seem to be changing, AI systems can be programmed to stay up to date with the latest changes and can be updated to ensure that the documents are automatically complying with new regulations. So it's entirely adaptable, and that's particularly beneficial if you're trying to coordinate global submissions. So that can be anything from the submissions under the IVDR or the FDA submissions, and figuring out ways of improving your processes so that you can align those two submissions and perhaps leverage some of the requirements across both submissions rather than redoing it from scratch and treating each submission as a standalone process. The other major advantage that we're seeing early on is this idea of scalability. So an AI system is able to really scale up to handle increases in data volume. And it's doing so without compromising the quality of the data that's being extracted. So as you are thinking about your overall processes from the writing perspective, think about developing sort of these databases that compile the results of all of these systematic literature reviews. That scalability will help you with these large scale projects where you may have multiple products as part of a larger product family, or if you're trying to coordinate and establish results from different clinical trials in different populations, and then assess that data and apply that data to stratify it based on which submission you're applying for. So these are just some of the key findings that we've identified from just sort of wandering around at DIA, listening to the various speakers, and also speaking with a lot of the folks there in attendance who are really struggling with sort of the relative roles of companion diagnostic partners as well as the pharma partners in this whole new process. We're all trying to leverage what limited data is available to us and trying to figure out the ways of building in efficiencies to ensure that both partners are able to coordinate their efforts and ensure the timeliness of these submissions. So I've built in a little bit of time here for some questions at the end, just in case anyone has some additional questions to better understand sort of the impact of these IVDR regulations and the new updated FDA regulations as they pertain to companion diagnostics and LDTs. So a big thank you to everyone who submitted questions ahead of time. But if you didn't have a chance to, or if you've thought of something, you can also feel free to use the Q&A button, and that can allow you to submit some questions live here.
Awesome. Thank you, Sarah. We will now move into our Q&A portion of the presentation. There were a few questions that were submitted with registrations and throughout today's presentation. However, please do not hesitate to submit your own using the Q&A button in the bottom menu bar, as Sarah just described. We will do our best to get through as many as possible. As always, if you run into any issues, please do not hesitate to send us a message, and we'll be happy to help troubleshoot. That said, let's get started with our first question. How can I leverage published literature to locate RWE in support of my CDX approval?
So this is really drilling down to the importance of designing a good search. The AI systems, I think, in my experience, people are relying on the AI to help them craft a way of extracting data from a very large data set. But if you can build the search very purposefully and very targeted approach to your search, that is going to help you really drill down and be more efficient in the overall process, even above and beyond what the AI is currently capable of. So I would say, your first step is going to be designing a very purpose-built search strategy. Because when you're looking at it from the perspective of a companion diagnostic, you are going to be identifying literature as it pertains to the test for that specific indication. So you're focusing in on the role of that IVD as it pertains to a particular drug partner or a pharma partner. And there is a tendency, if you search for just the drug partner or just the companion diagnostic, it may have other intended purposes outside the scope of your current review. And so in my experience, if you just use very limited search terms and it's a very high level search, you can be potentially looking at screening thousands of articles unnecessarily. But if you can work with a librarian to really craft the searches, to build in the correct syntax to allow you to identify the articles that have already been published with regards to that particular test in the context of informing treatment decisions, stratifying risk for patients. It's basically identifying a way of targeting your search more effectively. That I think is going to be your best strategy. You want to find a balance between making your search so specific that you don't actually identify all of the relevant literature that's out there, but also making it so broad that it's just an unmanageable amount of data to sift through. So it's finding a balance there and trying to figure out, is my search getting me the information that I need to support and establish scientific validity and state of the art?
Lovely. On to the next question. How would your approach change if the IVD is a novel device with limited clinical data published in the literature?
Yeah, I think this is really one of the main challenges we're having in the IVD space. When you're a medical device, it's assumed that by the time you get to clinical data, there have been clinical trials conducted. That's not always the case for IVDs. So in many cases, you may have an IVD that's been on the market for quite a while, and maybe you're refining your intended purpose or you're expanding your intended purpose. Maybe there isn't a whole lot of available options in the diagnostic space other than your device right now. And so the data may be very, very limited. I think in this case, it again, it goes back to your choice of your search terms and your PICO terms. So your population, your indications, comparators, and outcomes. If you can identify what are the alternatives if your device were not available, if your device were not on the market, what else would be available to individuals? And then show the potential clinical benefit for your device by adding it to the clinical pathways. That's going to help you establish that benefit and the clinical utility of your device. So don't underestimate the need to establish an unmet need and that clinical utility. The notified bodies are really looking for that in the event that you are one of these novel devices. You want to show the unmet clinical need. But you may have to also include some information pertaining to the preclinical data that shows that, yes, the clinical data is limited thus far, but the preclinical data supports it as being a state of the art or a new or emerging technology. So you may have to expand your searches to include some of the articles that are going to give you a really solid background for establishing scientific validity. So spending a little bit more time on your SVR compared to if it's a well-established technology. That would be my main approach I would take with a novel device.
Awesome. Thank you, Sarah. As a friendly reminder, you can submit any questions you have via the Q&A button on the Zoom menu bar. If you have more specific or confidential questions, you can always reach out to us directly at consult@criterionedge.com. Also, by attending this webinar, you are eligible for a free consult that can be reserved in the next 10 business days to discuss either a current or upcoming project you may have. If you wish to do so, feel free to pick a day/time that works best for you using the link in the chat. Or again, you can email us directly at consult@criterionedge.com, and we'll be happy to get something on the calendar. That said, let's move on to our next question. Do you have suggestions about how best to collect and present data on alternative or similar competitor devices?
This kind of addresses some of the potential of some of these AI technologies, really. I would say, starting this process very early, even if you have a device that's in development, the earlier you can start on collecting and identifying who are your most appropriate alternatives or competitors, the earlier you can start that process, the better. And I say that because you will find some of these alternatives or competitors that are starting to apply new information to their device design, and that could impact the decision making that your own research and development team is making. We've actually seen this quite a bit with some of our clients who come into the process, they have a device under development and they actually want to develop scientific validity um, early in the process. So they are not even to the market yet. And they need to know who are the alternatives out there, who are our competitors out there. And so you conduct a search early and you identify, well, according to the published literature, most of the other published data use this competitor of yours as a benchmark. And then you can compare yourself to that competitor. If it's a PCR based test, you can see, do you use similar primer sets? If it's if depending on the other alternative technologies that are out there, you can also look to see whether or not they're being used simultaneously as part of some of these diagnostic algorithms. Sometimes your test may be the confirmatory test, you may be the benchmark, and sometimes you are the comparator. You're being compared against a gold standard. And that's all really important to know early on. I would say my main suggestion as far as like the data presentation style is to clearly delineate between what is a competitor and what's an alternative. You wouldn't want to compare as you're developing your acceptance criteria, you wouldn't want to compare yourself against a gold standard benchmark that is an alternate type of technology if there are more similar competitor devices available. For instance, if you're with a very simple example like COVID testing, the sensitivity of a PCR based assay is going to be much different than serology. So, you want to understand what are the limitations of the technology and the platform that you're employing. If you are a novel device, then you may have to compare yourself against other alternatives. But if there are similar devices that have unique features that you could more closely compare yourself to, those would be the ones you would want to design your acceptance criteria to cover. So you want to make sure that you are comparing yourself to the right set of devices, so you don't set the bar too high or too low as far as what level of performance your device is expected to achieve to be able to be considered state of the art.
Excellent. Looks like we've got a few questions left. We'll keep moving on. If you have any questions you would like to submit, please feel free to do so using the Q&A option at the bottom. You can also refer to the links that are in the chat as well. That said, on to the next. For a new IVD development, how can the SVR be built to not only comply to IVDR, but to be of added value for the project decisions?
Yes, I think this is ultimately what the writers of the IVDR were intending in the first place. They put forth these extra added regulations, but the goal was to ensure that the products that are going to the market and that are being CE marked are of the highest quality and provide the best, most state of the art technologies available to us. So, as you're thinking about the SVR, don't just think of it as like a box to be checked, but it's also kind of that, market intelligence for understanding what are your competitors doing? Are they employing different technologies? If you're, again, with the COVID test example, if you are comparing yourself, are you able to properly identify new variants that are in circulation? If you're more of a, a genetic test, are you able to identify the variants that are most frequently seen in the European population? You have to take into account um, whether or not the choice of targets, the choice of biomarkers that you're employing will be considered state of the art by the time you get through the literature review process. If you can have a peek at that before you get too far along, it could actually impact your product development decisions early on. We've actually had several of our clients who have done an SVR, realized that all of their competitors have actually added a new capability that their particular device was unable to achieve. And so that actually led them to sort of take a step back and add additional primer sets, or identify maybe a new or emerging uh, antibiotic resistance or something like that that is being reported in the literature, so that they can better anticipate what will be state of the art. Will their device be able to meet that standard by the time they're ready to submit this requirement? So I would say, that scientific validity should be conducted early and often. You want to keep an eye out for what are the trends that you're seeing? Are there new regulations and recommendations that are being set out by reputable organizations? Are there different data or potentially cross reactivities, interferences that some of your competitors are reporting? Learn from their mistakes. If they're reporting that there's a new interference, that could be important information for you to know, so that you can build or design your test in such a way so that that is not a concern for you, and that sets you apart from your closest competitors. So, yeah, the SVR can be a really valuable tool, and I've seen it shared across not only the regulatory team but also with the R&D team. It's meant to be this feedback loop where you're conducting these searches regularly to be proactively identifying whether or not your device is still state of the art before it hits the market, but then also after it's already on the market. Does it continue to be state of the art?
Excellent. Looks like we've just got two left. Should the SVR be updated during the lifetime of the device as part of post-market surveillance activities?
Okay, absolutely. You want, and you may, some years, depending on the type of device you have, there may not be a lot of movement in the field from year over year. Like, for instance, if you have like an HIV test where you may have the same basic strategy and device configurations that have been on the market for decades, there may not be year to year movement, but you need to at least show that you're looking for any reports. So you still are expected to conduct these literature reviews and to be identifying any new potential concerns with the accuracy or the performance of the test as it's being published in real world data. So, that's where that real world data, sometimes you can't anticipate something until it's been published, or you find one of these case reports in the published literature. But you want to be able to show through your post-market surveillance activities that you are actively on the lookout for any potential reasons why your test may not perform as intended once it reaches the end user, the real world population. And also look for any instances where it's being used for off-market or off-label purposes. That's also important to know, even though that may not be within the scope of the SVR. It helps you identify maybe potential expansions for your intended purpose that you may wish to consider down the line, or maybe expanding to a different population. If your device is intended to be used in an adult population, but you're seeing published reports of it being used successfully in a pediatric population, it may be worth taking a look at whether or not your intended purpose could be expanded.
Lovely. And last but certainly not least, what are some simple ways to start preparing for submissions in the next 1 to 2 years?
For me, if I had a device and I was saying, I'm going to plan to submit under the IVDR in 2 years, I would start right away with a state-of-the-art search and a guideline search. Those would be the bare minimums I would start with. I would want to know what are the current recommendations, what are the current alternatives, for which populations is a device like mine intended to be used with. And also identifying things that I may not have anticipated, those off-label uses. So I would say that state-of-the-art search, that alternative search, and identifying any relevant clinical practice guidelines straight away. And then taking a look at the competitive landscape. If you can identify how others are assessing the performance of their tests, you may be able to build in the same types of standards or the same types of statistical analyses, to be able to make sure that the results that you obtain are comparable. I have seen this before where you may be collecting one piece of data, and the way your trial is designed, you maybe are using a different time point or a different endpoint than some of your comparators do. If you do a competitive landscape analysis, you'll know going into it that people are more typically assessing performance at these particular time intervals or in these particular sample types. You'll know that going into it so that you can better align your experiments with your competitors' published data to be able to give you that sort of apples to apples comparison when you go to extract the data and compile your landscape analysis. It just makes sure that there wasn't a piece of data that you could have caught. If you had known that others were reporting out that piece of data, you could have easily integrated that into your analytical performance plans from the get-go had you only known about it.
Excellent. And that is going to conclude our Q&A portion of today's webinar. Thank you to everybody that took the time to join us. We will be sending out a recorded version of this presentation in the coming week, so please keep an eye on your inboxes for that. As a reminder, if you would like to reach out to somebody from our team or schedule the free consult that you are eligible for the next 10 business days, you can refer to the links that are in the chat. The email is consult@criterionedge.com, and the meeting link can be found directly in the chat itself. Thanks again for everybody that joined. We will stay on for another 1 or 2 minutes for anybody else that has questions. Otherwise, we hope you have a wonderful rest of your day and a wonderful rest of your week.